So we are going to do Pseudomonas aeruginosa. Aeruginosa can give the hint to how it is aerobic, motile, catalase positive, gram-negative rod, non-lactose fermented, oxidase positive, frequently found in water, increased virulence in acidic environments, has a great odor. Pseudomonas is associated with E for pneumonia, S for sepsis, E for erythema gangrenosum, U for UTIs, D for diabetes, O for osteomyelitis, M for mucoid polysaccharide capsule, O for otitis externa, swimmer's ear, and N for nosocomial or healthcare associated infections, example catheters or equipment, A for addiction, such as injection drug use, and S for skin infections, example frontal folliculitis, worm infection in bird victims. So, what is erythema gangrenosum UTI? The erythema gangrenosum. Erythema gangrenosum. Oh, no. Erythema gangrenosum. I think my internet is slow. So we got to do this and then we need to. So it's erythema gangrenosum. And then we have UTIs, diabetes, osteomyelitis, mucoid polysaccharide capsule, otitis externa, nosocomial, addiction-related skin infections. It has a mucoid polysaccharide capsule that may contribute to chronic pneumonia in patients with CF due to biofilm formation. Produces PEEP, phospholipase C, that degrades the cell membranes, endotoxin that causes fever and shock, exotoxin A that inactivates EF2, and green pigment. That is shown in this diagram. Corneal ulcers or keratitis in contact lens wearers or minor eye trauma can also be caused by this bacteria. Erythema gangrenosum, okay, so it is written right here. It is rapidly progressive necrotic cutaneous lesion that is caused by Pseudomonas anemia, typically seen in immunocompromised patients. So it is a rapidly progressive necrotic cutaneous lesion. The treatments are anti-pseudomonal penicillins in combination with beta-lactamase inhibitor, example piperacillin and tazobactam. Third and fourth generation cephalosporins, examples of ceftazidime and cefepime, monobactams, fluoroquinolones, and carbapenems. Despite anti-pseudomonal activity, aminoglycoside monotherapy is avoided due to poor performance in acidic environments. And this one is increased virulence in acidic environment, has a grape-like odor, and thus aminoglycoside monotherapy is avoided, although it has anti-pseudomonal activity. Again, the treatments are beta-lactamase inhibitors with penicillins, third and fourth generation cephalosporins, monobactams, fluoroquinolones, and carbapenems. It produces PEEP, that is phospholipase C, endotoxin, fever and shock, azotoxin A, and green pigment. Next, we have Salmonella versus Shigella. Both Salmonella and Shigella are gram-negative rods that are non-lactose fermenters, oxidase negative, and can invade the GI tract via M cells of bile patches. Both of them can. Again, gram-negative rods, non-lactose fermenters, oxidase negative, and can invade the GI tract via M cells of bile patches. The first one is Salmonella typhi. The second one is Salmonella except Salmonella typhi, and then we have Shigella. The reservoir of Salmonella typhi is human only, and for other species, humans and animal, and Shigella is also in humans. Salmonella typhi has a hematogenous spread. And Salmonella except the typhi has a rare hematogenous spread. Shigella has cell-to-cell and there is no hematogenous spread. Salmonella typhi has H2S production. Salmonella except Salmonella typhi also have H2S production, but Shigella does not have H2S production. Salmonella typhi have flagella, and other Salmonella species also have flagella, but Shigella does not have flagella. The virulence factors are endotoxin and VI capsule, pronounced TIVI, TYVI, endotoxin and VI capsule. And the Salmonella species has endotoxin, and Shigella has also endotoxin called Shiga toxin, which is enterotoxin. So all of them have endotoxin, and the virulence factor of Salmonella typhi is also the VI capsule. Infectious dose or ID50 is very high for Salmonella typhi. Large inoculum required. It is acid labile, and thus it is inactivated by gastric acids. It is high for other Salmonella species as well, but for Shigella it is very low, very small inoculum is required. Acid stable as compared to Salmonella, and it is resistant to gastric acids. Effects of antibiotics on fecal excretion. So, if we give antibiotics to patients that are infected by these bacteria, uh, then for Salmonella typhi infection, fecal excretion is prolonged, and for Shigella, it is shortened. And for other Salmonella species, it is prolonged as well. Immune response of Salmonella typhi is by primarily monocytes, and mm, for Salmonella other species, it is PMNs in disseminated disease. Mm, I don't know what PMN is, but it is also present in Shigella. Then we have GI manifestations that are constipation followed by diarrhea for Salmonella typhi, diarrhea possibly bloody for Salmonella, except Salmonella typhi, and crampy abdominal pain that causes tenesmus, bloody mucoid stools, bacillary dysentery for Shigella. Then we have vaccine. For Salmonella typhi, oral vaccine contains live attenuated S. typhi. IM vaccine intramuscular contains VI capsular polysaccharide. No vaccine for other Salmonella species or Shigella. The unique properties of Salmonella typhi is that it causes typhoid fever, salmon-colored truncal macular rash, abdominal pain, fever, pulse temperature dissociation, later GI ulceration and hemorrhage, treat with ceftriaxone or fluoroquinolone and carrier state with gallbladder colonization. Okay, so a lot of important stuff. Truncal macular rash, pulse temperature dissociation, and it has this pulse which is called relative bradycardia, and there's also ulceration and hemorrhage in the GI and gallbladder colonization. In other salmonella species, there's poultry, eggs, pets, and turtles are common sources. Treatment is supportive. Antibiotics are not indicative in immunocompetent individuals. For Shigella, we have four F's: fingers, flies, food, and feces, in order of decreasing severity: S. dysentery, S. flexneri, S. boydii, and S. sonnei. Invasion of M cells is key to pathogenicity. Infectious dose is low. So a very small inoculum is required for Shigella to spread. Our next bacteria is Yersinia enterocolitica. It is a gram-negative pleomorphic rod or Cocobacillus with bipolar staining. It is usually transmitted from pet feces, example cats and dogs, contaminated milk or pork, can cause acute bloody diarrhea, pseudoappendicitis, right, lower abdominal pain due to mesenteric adenitis or terminal ileitis, and reactive arthritis in adults. Next, we have lactose fermenting enteric bacteria. So all of these bacteria were enteric bacteria, but they were not lactose fermenters. Right here. Then the bacilli that are not lactose fermenters uh, include those that are oxidase positive or negative. Oxidase positive are Pseudomonas and Burkholderia, and oxidase negative are those that produce H2S or those that do not. And this we've discovered all these lactose negative lactose non-fermenters. And now it is the turn of lactose fermenters, which are E. coli, Klebsiella, Enterobacter, Citrobacter, and Serratia. So the first one is, okay, lactose fermenting enteric bacteria. They are fermenter of lactose that form pink colonies on McConkey agar. Examples include Citrobacter, E. coli, Enterobacter, and Klebsiella. McConkey keeks milk. Cow for Conkey, keeks for Citrobacter, E. coli, Enterobacter, and Klebsiella. EMB agar is lactose fermenter grow as purple or black colonies on these EMB agars. E. coli grows colonies with green shell. Okay. The first one is E. coli. It is a gram-negative indole positive rod. E. coli virulence factors are the Fimria, cystitis that causes cystitis and pyelonephritis. There is K capsule that causes pneumonia, neonatal meningitis, and LPS endotoxin that causes septic shock. Again, important virulence factors: Fimria that causes cystitis and pyelonephritis, and K capsule that causes pneumonia, neonatal meningitis, and LPS endotoxin that causes septic shock. So there are different strains: entero-invasive E. coli, enterotoxigenic E. coli, enteropathogenic E. coli, and enterohemorrhagic E. coli. in those that invade, those that produce toxic effects, those that cause pathogenicity, and those that cause hemorrhage. Enteroinvasive E. coli are microbes invade intestinal mucosa and cause necrosis and inflammation. Uh, there's dysentery, clinical manifestation similar to Shigella. Then there's enterotoxigenic E. coli that produces heat labile and heat stable enterotoxins. These were the invaders and they were causing necrosis inflammation. These are producing enterotoxins that are heat labile and heat stable, and there's no inflammation or invasion. And they are causing traveler's diarrhea, which is watery diarrhea. The invaders were causing diarrhea that was similar to Shigella or dysentery. Then we have enteropathogenic that produce no toxins. They adhere to apical surface. They flatten the villi and they prevent absorption. They just are pathogenic. They cause diarrhea usually in children, think EPIC and pediatrics. Then we have enterohemorrhagic E. coli that is O15787 is most common serotype in the US, often transmitted by undercooked meat and raw leafy vegetables. Shiga toxins causes hemolytic uremic syndrome, which is a triad of anemia, thrombocytopenia, and acute kidney injury due to microthrombi forming on damaged endothelium. And this can cause mechanical hemolysis with schistocytes in peripheral blood smear and platelet consumption. And it can also lead to thrombocytopenia and decreased renal blood flow. This causes dysentery. It does not ferment sorbitol versus other E. coli, and EHEC associated with hemorrhage, hamburgers, and hemolytic uremic syndrome. So it is associated with hemolytic uremic syndrome, which is essentially anemia, thrombocytopenia, and acute kidney injury. It is associated with mechanical hemolysis, that is hemorrhage with schistocytes, and it is associated with thrombocytopenia and decreased renal blood flow. An important property of this is that it does not ferment sorbitol versus other E. coli. Okay. Then we have Klebsiella, that is a gram-negative rod intestinal microbiota that causes lobar pneumonia, more common in patients with heavy alcohol use or period post offenses. It has very mucoid colonies as shown in this picture, caused by abundant polysaccharide capsule, dark red currant jelly sputum, also cause of healthcare associated UTIs associated with evolution of multi-drug resistance. ABCDs of Klebsiella are aspiration pneumonia, abscess in the lungs and liver, currant jelly sputum, diabetes mellitus, and ETOH overuse. Next one is Campylobacter jejuni. It is gram-negative, comma, or S-shaped with polar flagella. It is oxidase positive, grows at 42 degrees Celsius. Campylobacter likes the hot campfire. Major cause of bloody diarrhea, especially in children. Fecal oral transmission through person-to-person contact or via ingestion of undercooked, contaminated poultry or meat, unpasteurized milk. Contact with infected animals such as dogs, cats, pigs is also a risk factor and common antecedent to Guillain-Barré syndrome and reactive arthritis. I wish I had more internet to search Guillain-Barré syndrome, but yeah. Bloody diarrhea, undercooked contaminated poultry or meat, and pasteurized milk. Among all of the bacteria that we've discussed, these were non-lactose fermenters and they were causing diarrhea, and the Shiga toxin was causing dysentery, which is bloody mucoid stools, and Yersinia was causing pseudoappendicitis. E. coli were causing diarrhea of all the types, watery diarrhea in children and dysentery, and depending on the type, and Klebsiella is again causing aspiration pneumonia and abscess in the lungs. Campylobacter jejuni is causing bloody diarrhea. Next, we have Proteus mirabilis. It is a gram-negative urease positive facultative anaerobe that has long flagella with swarm motility. So it is a facultative anaerobe with long flagella and swarm motility. Common causes of UTIs: urease hydrolyzes urea to carbon dioxide and ammonia. There is net increase in pH and promotes the formation of struvite stones. Significant blockage of renal calyces results in branch stones called staghorn calculi. Next, we have Vibrio cholera, that is a gram-negative flagellated comma-shaped oxidase-positive bacteria that is grown in alkaline media. The one that was grown in acidic media was, I'll have to look up. I keep forgetting these all the time. The one that grows in acidic media was Pseudomonas, okay. And this one requires an alkaline media and it is Vibrio cholera. It is endemic to developing countries. It produces profuse rice water diarrhea by enterotoxin that permanently activates GS. It increases cyclic AMP. It is sensitive to stomach acid, so it is acid labile and requires large inoculum, just like salmonella. And unless host has decreased gastric acidity, transmitted by ingestion of contaminated water or uncooked food, example raw shellfish, it is treated promptly with oral rehydration solution. Then we have, okay, so oral rehydration solution is very important in Vibrio cholera. Then we have Vibrio vulnificus. It is a gram-negative bacillus that is usually found in marine environments. It causes severe wound infections or septicemia due to exposure to contaminated seawater. So it is also associated with water in some way, right? It presents as cellulitis that can progress to necrotizing fasciitis in high-risk patients, especially those with high serum iron, example cirrhosis and hemochromatosis. It is serious wound infection requires surgical debridement. Okay. So what is important about this one is that it presents a cellulitis that can progress to necrotizing fasciitis in high-risk patients, especially those with high serum iron. Then we have an Helicobacter pylori that is curved, flagellated, motile, gram-negative rod that is triple positive. It is catalase positive, oxidase positive, and urease positive. It can cause urea blood test or fecal antigen test for diagnosis. Urease produces ammonia, creating an alkaline environment, which helps H. pylori survive in acidic mucosa. It colonizes mainly the antrum of stomach, causes gastritis and peptic ulcers. Risk factor for peptic ulcer disease, gastric adenocarcinoma, and mild lymphoma. Okay. Most common initial therapy is triple therapy, that is amoxicillin, metronidazole if there is penicillin allergy, clazithromycin, and PPIs. Antibiotics cure pylori. Bismuth-based quadruple therapy if concerned about macrolide resistance. So amoxicillin, if there is penicillin allergy, then you go metronidazole along with clarithromycin and PPIs. And for quadruple therapy, you use bismuth-based if you're concerned about macrolide resistance. The next group is spirochetes that are spiral-shaped bacteria with axial filaments, include Leptospira, Treponema, and Borrelia. Only Borrelia can be visualized using aniline dyes, right? Teams are stain in light microscopy due to size. Treponema is visualized by dark field microscopy or direct fluorescent antibody or DFA microscopy. I again wish I had better internet so I could search what DFA was, but I think it is worth a try if I. Oh yes. Um, um, A direct fluorescent antibody test is a rapid immunofluorescence assay that uses a single fluorescein labeled monoclonal antibody to detect specific microbial antigens directly in patient specimens, producing an apple green fluorescence under a microscope. And this is helpful in detecting treponema. Then we have Jerisch-Herxheimer reaction, that is flu-like symptoms, fever, chills, headache, myalgia after antibiotics are started due to host response to sudden release of bacterial antigens. It usually occurs during the treatment of spirochetal infections. So spirochetes, again, only Borrelia can be visualized using aniline dyes, and treponema is visualized by dark field or DFA. First is Lyme disease. It is caused by Borrelia burgdorferi, which is transmitted by the Ixodes layer tick, which is here. It is also a vector for Anaplasma and Protozoa vivacia. Natural reservoir is the mouse. Deer are essential to take life cycle but do not harbor Borrelia. They are basically for the adult reservoir. Common in northeastern United States, stage 1 is early localized erythema migrans that is typically bull's eye configuration right here. It is pathognomic but not always present. Flu-like symptoms appear in this stage. Stage 2 is early disseminated secondary lesions, carditis, AV block, facial nerve palsy, migrated myalgias, and transient arthritis. Stage 3 is late disseminated encephalopathy, chronic arthritis, and peripheral neuropathy. A key line pie to the face is facial nerve palsy, erythritis, cardiac block, and erythema migrans. Treatment is doxycycline, first line, and then there's amoxicillin in pregnant patients and children less than 8 years of age, and then there's ceftriaxone if IV therapy is required. So first line is doxycycline. For pregnant patients and children, there's amoxicillin and ceftriaxone if IV therapy is required. I'd like to switch erythema migrans. Erythema migrans, yes. This one was erythema. The one we did earlier was uh, erythema erythema gangrenosum for Pseudomonas aeruginosa, and this one is erythema migrans, right here. It is an expanding red or bluish rash, often the first sign of Lyme disease, is appearing at the site of tick bite within days to weeks. Okay, so it is specific to Lyme disease. Next one, we have Leptospira interrogans. It is a spirochete with hook-shaped ends found on water contaminated with animal urine. It causes leptospirosis and wheal disease. Leptospirosis is flu-like symptom, myalgias, jaundice, photophobia with conjunctival fusion, prevalent among surfers and in tropics. Wheal disease is pterohemorrhagic leptospirosis. Okay. It is severe form of jaundice and azotemia from liver and kidney dysfunction, fever, hemorrhage, and anemia. So basically, just two diseases: leptospirosis and whale disease. Leptospirosis has flu-like symptoms and myalgias, conjunctival fusion, and whale disease has jaundice and azotemia from liver and kidney dysfunction. Then we have syphilis, which is caused by the spirochete Treponema pallidum. Its treatment is penicillin G, which is very high yield. We have primary syphilis, that is localized disease presenting with painless chancre. They use fluorescent or dark field microscopy to visualize treponemas in fluid from chancre. The VDRL is positive in approximately 80% of the patients. Then we have secondary syph, which is a disseminated disease with constitution symptoms, including maculopapular rash, including palms and sole. This one. Then there's condyloma letta. Wait, uh, palms and soles. This one is the rash, maculopapular rash, all C1, C2, C3. And D is the condyloma letta, which is smooth, painless, wart-like white lesions on the genitals. Then we have lymphadenopathy, patchy hair loss, also confirmable with dark field microscopy. Serologic testing is VDRL or RPR, which are non-specific, and the diagnosis is confirmed by specific tests such as FTA and ABS. Secondary syph is systemic. Latent syphilis may follow, which has positive serology without any symptoms. So VDRL is positive in 80% of the patients, and secondary syph can only be confirmed with FTA ABS. Then we have tertiary syph, gammas, that is chronic granulomas, which are shown in this diagram. These right here. Aortitis, vena vasorum destruction, neurosyphilis, stapes dorsalis, and general paresis. Arthur Robertson pupil constricts with accommodation but is not reactive to light. Signs are broad-based ataxia, positive rhomboid, Charcot joint, and stroke without hypertension. Broad-based ataxia, positive rhomboid, Charcot joint, and stroke without hypertension. Okay. So what is broad-based ataxia? It is a type of unsteady white stance gait caused by dysfunction in the cerebellum, vestibular system, or sensory pathways. Broad-based ataxia is a type of unsteady white stance gait caused by dysfunction in the cerebellum, vestibular system, or sensory pathways. Okay, so it is affecting that, and then there's a positive rhomboid test. Positive rhomboid test. That one is indicates a loss of balance when the eyes are closed, suggesting a cerebellar problem basically. In the Charcot joint. Charcot joint. A neuropathic arthropathy, progressive destructive joint disorder caused by loss of sensation. Okay, so it is basically a problem with joints, but because of your nervous system. Then we have congenital syph that presents with facial abnormalities such as rigidez, which are linear scars at angles of mouth, and black arrow in F. Okay, so this is what a rigidez is. Snuffles, nasal discharge, right here. Saddle nose, notched or Hutchinson teeth, such as shown right here. Mulberry molars and short maxilla. Saber shins, cranial nerve number 8 deafness. To prevent treat patient early in pregnancy as placental transmission typically occurs after first trimester. Okay, so now we are up to diagnosing SIF. How much are we left with? Okay, you'll just diagnose it for now. VDRL and RPR detects non-specific antibody that reacts with beef cardiolipin. Quantitative, inexpensive, and widely available test for safe, sensitive but not specific. Non-treponemal tests, VDRL, RPR, and direct testing revert to negative after treatment. Antibodies detected by treponemal test, FTA, ABS, or TPA-PA will remain positive. Okay, how about we just first do? The direct testing is by dark field and PCR. Serologic testing might be non-treponemal or treponemal. And the non-treponemal test will react with non-specific antibody. They are inexpensive and widely used, but they're not specific. And they revert to negative after treatment. False positive test of VDRL will be given in pregnancy, uh, viral infections, for example, in EBV and hepatitis, drugs such as chlorpromazine and procainamide, rheumatic fever, which is rare, and lupus, anticardiolipin antibody, and leprosy. So these are all the causes in which we are going to have false positive tests. So you better confirm it with treponemal specific, which are FDA, ABS, and DPPA. Next is chlamydia. Chlamydia cannot make their own ATP. They are obligate intracellular organisms that cause mucosal infections. They have two forms. The elementary body is small and dense. It is infectious and enters the cell via endocytosis. It transforms into a reticulate body. The reticulate body then replicates in cell by fission and reorganizes into elementary bodies. Okay, so elementary body, then reticulate, and then again there is elementary body. Chlamydia trachomatis causes neonatal and follicular adult conjunctivitis. It also causes non-gonococcal urethritis and PID and reactive arthritis. Okay, it is going to cause conjunctivitis, urethritis, which is non-gonococcal, and PID and reactive arthritis. Chlamydophila pneumonia and Chlamydophila psittaci cause atypical pneumonia, which is transmitted by aerosol. So trachomatis is going to cause the conjunctivitis, urethritis, and chlamydophila are going to cause pneumonia, and this pneumonia is transmitted by aerosol. Chlamydial cell wall lacks classic peptidoglycan due to reduced muramic acid, rendering beta-lactam antibiotics ineffective. Chlamys means clog, that means intracellular. C. Psittaci has an avian reservoir, which means parrots that cause atypical pneumonia, and lab diagnosis is by PCR, NAT, cytoplasmic inclusions seen on GIMSA or fluorescent antibody stained hairs. Treatment is by doxycycline, azithromycin in pregnant patients, adcetrizone if concurrent gonorrhea test is positive. We also discussed this in gonorrhea. If when we were treating gonorrhea, if there is also chlamydial infection, then you add ceftriaxone, and if there was no chlamydial infection, then you could go with doxycycline alone, and you can go with doxycycline alone in case of chlamydia as well. Chlamydia trachomatis has 15 serotypes. Types A, B, and C cause chronic infection that causes blindness due to follicular conjunctivitis in resource limited areas. A is for Africa, B is for blindness, and C is for chronic infection. What is follicular conjunctivitis? Follicular conjunctivitis. It is an inflammation of the conjunctiva characterized by swollen immune follicles, usually caused by viral or chlamydial infections. Okay. Then we have type B to K that causes urethritis, ectopic pregnancy, neonatal pneumonia or staccato cough with eosinophilia, neonatal conjunctivitis 1 to 2 weeks after birth. B2K is for everything else. Neonatal disease can be acquired during vaginal birth if pregnant patient is infected. So yeah, neonatal pneumonia, eosinophilia, and neonatal conjunctivitis. Types L1, L2, L3 cause lymphogranuloma venereum, which is small painless ulcers on genitals that are swollen, painful inguinal lymph nodes that ulcerate and treat with doxycycline. Next, we have Gardnerella vaginalis, which is a pleomorphic gram-variable rod involved in bacterial vaginosis. It presents as a gray vaginal discharge with a fishy smell. It is non-painful versus vaginitis, which is painful. This is a very important feature of it. Vaginitis is going to be painful, but this is not painful. It is associated with sexual activity, but not sexually transmitted. Bacterial vaginosis is also characterized by overgrowth of certain anaerobic bacteria in the vagina due to decreased lactobacilli. Clue cells, which are vaginal epithelial cells covered with Gardnerella, have stipple appearance along outer margin. So these are the clue cells right here. There's an amine BIF test, which is mixing discharge with 10% KOH that enhances the fish odor, and the vaginal pH, if it is greater than 4.5 during infection, it indicates the presence of Gardnerella vaginalis because then there's going to be decreased lactobacilli. And it is treated with metronidazole and clindamycin. Again, it has a gray vaginal discharge and a fishy smell and gram-invariable voids, which are pleomorphic. Then we have zoonotic bacteria. Zoonosis is an infectious disease transmitted between animals and humans. These are anaplasma causing anaplasmosis by exodistic, Bartonella that causes cat scratch disease or bacillary angiomatosis, and it is spread by cat scratch. What is bacillary angiomatosis? I want to see the images. Okay, so this is what it looks like. It is a vascular lesion that can appear on the skin and other organs. Right. Then we have Borrelia burgdorferi that causes Lyme disease. We have already done that. Exodistic. Then we have Borrelia recurrentis that causes relapsing fever because it is recurrent and it is because of louse. Then we have brucella that causes brucellosis or unurine fever. It is present in unpasteurized dairy, inhalation of or contact with infected animal tissue or fluid. Then we have Campylobacter that causes bloody diarrhea, feces from infected pets, animals, contaminated meat, food, and hands. Then we have Chlamydophila cetaceae that causes psittacosis, or however it is pronounced, and it is spread by parrots and other birds. We have Coxiella burnetii that causes Q fever, and it is from the aerosol of cattle, sheep, or amniotic fluid. Then we have Ehrlichia sheppiensis that causes ehrlichiosis, and it is spread by emblyoma, which is a lone start. This is a new one for me. Then we have Francisella tularensis that causes tularemia and it is spread by ticks, rabbits, tail flies. Leptospira that causes leptospirosis and it is spread by animal urine in water and recreational water use. Mycobacterium lepra that causes leprosy and is spread by humans with leprosy and armadillo. Then we have Pasteurella miltosida that causes cellulitis and osteomyelitis and it is spread by animal bite, cats, and dogs. We have Rickettsia provazeki that causes epidemic typhus and it has human to human via human body louse. We have Rickettsia rickettsii that causes Rocky Mountain spotted fever and it is spread by dermacentor. We have Rickettsia typhi that causes endemic typhus. So the one that has typhi in it causes endemic typhus and the one that has provazeki in it causes epidemic typhus. And it is spread human to human, but endemic typhus is spread by fleas. Then Salmonella species, except typhi, causes diarrhea, vomiting, fever, abdominal cramps. It is spread by reptiles and poultry, and Yersinia pestis causes a plague, and it is spread by fleas, rats, and prairie dogs are the reservoirs. Then we have rickettsial diseases and vector-borne illnesses, which are, I think, yes, the last in bacteriology, very fortunately. So rickettsial disease and vector-borne illnesses are treated by doxycycline. That is one establishment we are going to develop. The rash that is common is Rocky Mountain fever and typhus. And the ones that are rare are ehrlichiosis, Q fever, and anaplasmosis. So with the common rashes, we have Rocky Mountain spotted fever that is caused by Rickettsia rickettsii. The vector is a tick. Despite its name, disease occurs primarily in the South Atlantic states, especially North Carolina, however it's pronounced. Rash typically starts at wrists and ankles and then spreads to trunk, palms, and sole. There is a classic triad of fever, headache, and rash. Palms and sole rash is seen in Coxsackie virus A infection, hand, foot, and mouth disease. Rocky Mountain spotted fever and secondary syphilis, you drive cars using your palms and soles. Okay, so this classic triad of headache, fever, and rash is seen in Coxsackie virus A infection, Rocky Mountain spotted fever because of rickettsii, and because of secondary syphilis. Few of these diseases. Typhus is endemic, that is caused by Rickettsia typhi, and epidemic, that is caused by human body louse. And this one is caused by Rickettsia prowazekii. Rash starts centrally and spreads out, spraying palms and soles. So the Rocky Mountain spotted fever essentially has the palms and soles, but typhus essentially does not have the palms and soles. There's rickettsia on the wrists, typhus on the trunk. Rickettsia on the wrist and typhus is on the trunk, sparing the palms and soles, and it may be endemic or epidemic, endemic by I. typhi and epidemic by I. prowazekii. Then we have weird rashes such as ehrlichiosis. Ehrlichia vector is tick, monocytes with morula or mulberry-like inclusions in the cytoplasm. This one. Ehrlichia vector is a tick, and monocytes with morula, mulberry-like lesions right here in the cytoplasm are the characteristic features of ehrlichiosis. Then we have anaplasmosis. Anaplasma vectoristic granulocytes with morula and cytoplasm. So this one has granulocytes in the cytoplasm, anaplasmosis. Mega, M for ehrlichiosis, monocytes, and G for anaplasmosis, granulocytes. Then we have Q fever that is caused by Coxiella burnetii. There is no arthropod vector. Bacterium inhaled as aerosols from cattle or sheep amniotic fluid. Presents with headache, cough, flu-like symptoms, pneumonia, possibly in combination with hepatitis. This is an important distinction. Common cause of culture is negative endocarditis. Q fever is caused by a quite complicated bug because it has no rash or vector and its causative organism can survive outside in its own form, not in Rickettsia genus, but closely related. So we add it here. There's no endocarditis. And the last one is Mycoplasma pneumoniae. It is a classical cause of atypical walking pneumonia, insidious onset, headache, non-productive cough, patchy or diffuse interstitial infiltrate, and macular rash. It occurs frequently in those less than 30 years of age, outbreaks in military recruits, prisons, and colleges. The treatment is macrolides, doxycycline, or fluoroquinolone. Penicillin is ineffective since mycoplasma has no cell wall. It is not seen on gram stain. It is pleomorphic. Um, where's the picture? Oh, there's the picture. It is pleomorphic. Bacterial membrane contains sterols for stability, and it is grown on etonic. Chest X-ray appears more severe than patient presentation. Higher titer of cold agglutinins IgM, which can agglutinate IBCs. Mycoplasma gets cold without a coat, no cell wall. And we are done with our bacteria. And I'm so happy that they're finally over. See you in the next video, hopefully. Bye.